Hi, in this video we're going to compare DNA and protein sequences using various sequence alignment algorithms from the bioinformatics toolbox of MATLAB. The comparison is going to be between a human and a mouse, which is the model organism, and we are going to compare the DNA that encodes the enzyme hex A, and that enzyme is responsible for the breakdown of GM2 gangliosides in the brain and nerve cells. Without that process happening, a person gets the Tay-Sachs disease, which causes paralysis, dementia, blindness, and early death on human beings. Okay, so let's start this example. The first step is going to the NCBI database and search for the Tay-Sachs disease. And that way we're going to find the corresponding gene. So if we search for Tay-Sachs, and get the first one, we get this entry, and at the bottom you get the sequence for the DNA. And okay, so this should be the corresponding, this is like the ID for the entry, and there's a command in MATLAB to pull all this information. We're gonna do that, but to be aligned with the article, I'm gonna use the code that they have, which is this one. I think it mentions here as well. So let's search for it in the database. Okay, so we got it, and the DNA should be at the bottom. Yep, there it is. Okay, so we're gonna pull it with this command, get gene bank with the ID. Okay, let's try it out. So in that we got all the information that we found in the CBI database, the ID, locus name, the description, and if we have the variable now in the workspace. So if we want to see the sequence, the DNA sequence directly, we can do it like this, and we can see that it has a total of 4,785 characters. Okay, so we're gonna do the same for the mouse. We go also to NCBI database and we search for it. Don't think it mentions how exactly to find it, but this is the code for it. Yeah, so this should be for the mouse, and as you can imagine, the DNA sequence is going to be shorter, and that's what we're going to compare against. We're going to use the same command with the ID of the mouse gene, and we got a similar structure. It's the same structure but different values, and now we have it in the base workspace. Okay, so the next step is finding out the ORFs, open reading frames. That uses start codons and end codons to find the different segments. For example, here's the biggest one. We're going to see it directly. We're going to use the command sequence show ORFs, and we're going to apply both to the human and the mouse. Okay, let's use the command. So we use it on the sequence of the human gene, and now with this we get we have a total of 3 frames. The first one has about maybe 7 different open reading frames, and this is the biggest one. The last one, I think it has 3. You can see that they are green, they're shorter, and the middle one, they are not that big, but it has also maybe 7 or something like that. Okay, notice also that these open reading frames start with a 3-letter codon, ATG, and end with TGA, and the last ending codon is not part of the sequence. Sorry, in this case will be TGA here, and it starts in ATG, finishes in TGA, and you can see that in this table. This is an RNA based table, and this is DNA, so the T becomes a U in the RNA. So the first one is AUG, so that AUG, which is the start, yeah, that should be the start, and then the ending will be TGA, which UGA, so that's the stop. So that's the start and the end. So when you execute that command, we're gonna get the start and end of those open reading frames. So let's take a look at how they look. Let's take the first frame, and we can see that this is the initial one and the ending as we saw. The first one is the longest one. This has about maybe 1600 characters, but the other one maybe have 10 or something like that. They are pretty small. Start, end, start, end, start, end, start, end. And the final one, final frame, had only 3, and as you can see, they are short as well. Okay, so let's do now the same for the mouse. So we're going to execute the same command for the mouse, which is this one. We're gonna already have the mouse sequence in the workspace. And again, the first sequence, ATG, finishes in TGA, that's the biggest one. And notice that the second sequence in the first frame starts with an ATG, but it doesn't end. So we're going to see how that affects. And the final one, final third frame, there's no ORF. Okay, so let's see how that affects. Let's take the mouse, the first one. You can see that the first one has 1600 characters, and the second ORF doesn't have an ending because it started but it didn't have an end. Okay, and the third one, you can imagine that it's empty, as we saw. Notice that the first frame, the first open reading frame, is the largest one, the same thing as the human, so this will be the place where we're gonna get our match, and we're gonna see it later. Okay, so let's go to the next step. Now we're gonna compare them, but we're gonna compare first, we're gonna compare the proteins. Okay, so we have the DNAs, and so you're gonna use the nucleotide to amino acid function to go from DNA to protein. The cells to go to a protein have to go through RNA, and then and the RNA is used to assemble the protein. But since we're only working with information, we can go straight from the DNA to the protein. Okay, so let's use this function to get the protein for the human gene. Okay. Okay, this is the corresponding human sequence. 3 characters of a DNA encode for 1 character of a protein. So let's you can verify it like this. Yeah, so this one is 3 times larger than the other one, and you can see it in here. Okay. Okay, now let's also take the protein for the mouse. And the proteins have their subsequences that start with M and end with star. You will see some stars around here. For example, this one. This is an ending token. Okay. Now let's compare the human protein with the mouse protein, and we're gonna do it like this with this function. Okay, but before going there, this is the mouse protein, the human protein, and this is the window and the number of characters within the window that should match in order to get a match. So okay, so this could be visualized better like this. Okay, so what this is gonna do, it is gonna take a window of 4 and then takes a window and compare it against all possible windows of 4 in here. And if there are 3 characters matching, for example, let's say that we have ABCD and then we have ABXD, then this will match because we have 3 characters in common. So that window is going to be slide through all the possibilities in here, and then after finishing here, then it's going to go to the next window here and compare it and go and keep going with 4 windows and compare it with all 4 windows. So it's going to try all possibilities. And now with that, and notice another thing, that the length of the human protein is around 1600, and the mouse protein is around 600, a little smaller. So that corresponds to the indices that we get in here. Notice that we get almost 1600. This is the human index, the index for the human protein, and this is the index for the mouse proteins, around 600. And this is where the corresponding index matches, and we see that a lot of dark, so it matches at the right places. There are other matches sporadic, but those doesn't matter. We got our matching here. Okay, so that's one thing. And now we're gonna try another method. So now we're gonna use the Needleman-Wunsch algorithm to compare the two proteins. So let's just give it a try. Okay. Okay, let's show the alignment using this command. Okay, so that's gonna give us the alignment. This function is gonna be removed in a future release, and I think the recommended one is this one, sequence align viewer. We can give it a try. I don't know if that's available in the release that I have. You can see that it's comparing. The vertical bars are where there's a match, others are mismatches, and we can see that there's a lot of matches at the beginning. And notice that we get dashes to basically stretch one sequence and stretch the other. The top one, I understand that is the human one, and the other one is the mouse. So after some point, you can see the human sequence protein, I mean, is much more larger than the mouse, so at some point there's no match at all. So the first step for this is we're gonna trim it down. Notice that the percentages are pretty bad in here. So let's cut the sequence to see if we have better results. Okay, to cut the sequences, we're gonna use the start and ending characters in the protein, which is M start and start ending. So in order to get those indexes, we're gonna use this vectorized expression, which is basically comparing all the protein characters, all the amino acid characters within the protein against the ending character. So this is a vector of characters, and with this scalar comparison, that is a vectorized operation, it's gonna return a vectorized type with Boolean type, is corresponding to 1 if it is a star and 0 false if it is not. Okay, so the find command, what it's gonna do is take a Boolean vector and it's gonna return the indexes for the elements within that vector in which the value is true. So effectively, we get the indexes of the protein where we have the star character. And we're going to do the same for the mouse and also for the M character. So let's take that. Actually, we don't need to store it, so I'm not going to use a variable for it. Let's get the human start and, sorry, let's go back. Let's get the start and the end. Okay, so the first sequence, you can see that is the longest one, or at least one of the longest ones. So we're gonna take the first one that starts in character 15 and ends in 544. Okay, so let's get the open reading frame of the protein for that, corresponding to that. So it was 14 and 5. Whoops, we don't need human stuff. You can just put 544. Okay, so it starts. Okay, let me get it again. Whoops, it was 15. Okay, 15 and then at 554. Okay, now it makes sense. It starts with M and should end with star. Perfect. Okay, so we have the corresponding, the first protein, the long one for the human. We're gonna do the same for the mouse. Okay, and this more smaller supposed to be. Okay, so the first one is 11 and 539. So that's what we're gonna do. We're gonna take the mouse ORF, take the command from here. So that will be 11 and 539. Okay, starts with M, ends with star perfectly. And now we're gonna use the same algorithm. Now we have those variables in the workspace, so we're gonna execute the same algorithm again, but with the ORF that we obtained. Okay, now we see that we get a better alignment. There are more matches, so let's take a look with the show alignment command. Okay, I'm just using the wrong name. It's supposed to be global alignment. Okay, so now we got 95% match, which is way much better. You can see the matching there. So that was one way of doing it. Another way is instead of trimming and comparing the protein, we can use the overrides for the DNA that we got before, and we get the start and stop. The first one, which if you remember, they are the longest sequence, which most likely are going to have a match with the mouse, so we're going to do the same with the mouse. And then once we get that, get the we use the RFs to get the indexes, and from the index we get the subsequence in the DNA, and then convert the DNA to a protein, and then do the same algorithm for alignment using those protein ORFs. Okay, so let's give it a shot. Okay, let's make sure that we have the corresponding indexes ready in the workspace. Start and no, stop. Okay, so now we're gonna get the sequence. Okay, starts with ATG. Okay, now let's convert it to a protein. Okay, that's the corresponding protein. Okay, the same for the mouse. 31. Okay, now let's get the DNA for it. This is the DNA for it. And now let's convert that DNA to a protein. Okay, now that we have the protein, let's take a look at how this looks with the protein ORFs, and it looks good. So effectively, the first DNA ORF that we got had a very high matching, as we can see in here. So let's confirm that with the show alignment method. Okay, we can see that we got even a higher match, 95%, and that was easier. Okay. Okay, as this one is suggests, this function is going to be removed, and actually I think in the new MATLAB release is gone. So the alternative is using sequence alignment viewer, and if you try it, you get a better display with the matches. Okay. Okay, now let's try another alignment alternative, which is the Smith-Waterman algorithm. It basically takes the whole protein, and if you remember when we tried the whole protein last time, we got a very bad alignment. So we're gonna do exactly the same, but with this other algorithm, the Smith-Waterman. So we're gonna replace the name of the function. Okay, let's see how it goes. And you can see that we don't have this huge mismatch. It found automatically, so we didn't have to get into the trouble of neither trimming the DNA nor trimming the protein. It just found automatically the highest match. So it looks like this might be the best strategy offered for alignment. Okay, so now let's take a look at how good did it fare. Again, 94%. I mean, by doing the nucleotide comparison and then converted to protein, gave us a 95%. So it gave us a little bit better. But at least we didn't have to do any work for this Smith-Waterman option. Okay, so that's it. Thank you very much for watching.